diabetes. I'm Greg Berry, Assistant Director in the UAB Office of Alumni Affairs. Since this is a virtual discussion, please don't be surprised if there are any technical issues or if anything unusual is seen or heard throughout the evening. If your video freezes, click the reconnect button at the top of your screen. This will get you back to the video very quickly. Throughout tonight's presentation, be sure to submit your questions in the chat box. We will make sure our presenters have the opportunity to answer those questions. So tonight we are very excited to welcome two individuals for this critical Conversation about diabetes, Dr. Anath Shalev and Dr. Fernando Ovalle. Dr. Shalev is the director of the UAB Comprehensive Diabetes Center and a professor in the UAB School of Medicine in the Division of Endocrinology, Diabetes, and Metabolism. She got her education at the University of Basel Medical School in Switzerland. Her research interests include molecular biology of diabetes, pancreatic beta cells, and diabetes drug discovery. Dr. Ovalle is a professor of medicine and director of the Division of Endocrinology, Diabetes, and Metabolism in the UAB School of Medicine. He also serves as director of the Multidisciplinary Comprehensive Diabetes Clinic. He obtained his MD from the Universidad Autónoma de Nuevo León in Monterrey, Mexico. Dr. Ovalle, I hope I got that all right. So if you have any questions tonight, please go ahead and put those in the chat room. After the doctors speak, we will continue the conversation with those questions. At this point, I would like to welcome Dr. Shalev and Dr. Ovalle for their presentation. Guys, it's all yours. I know, Dr. Shalev, you're going to get your slideshow going, but I will turn things over to both of you.
Great. Thank you so much for the introduction, and I hope that everyone can see my slides. So again, we're going to try to discuss novel approaches to diabetes. And first of all, I just want to kind of give you a little bit the numbers about the second pandemic, the diabetes pandemic. So diabetes has become a worldwide epidemic affecting 463 million people worldwide. And even by 2045, it is expected that 700 million will be affected. So it's really increasing in its extension. Over 30 million Americans currently suffer from diabetes, and over 100 million Americans have diabetes or prediabetes, so are at risk at developing diabetes down the road. And in fact, an additional 1.5 million Americans are diagnosed with diabetes every single year. So it's really a massive expansion of the disease. Now, here in Alabama, the prevalence of diabetes is really high, with over 15% of adults and over 20% in some counties. Currently, obviously, diabetes has also emerged as one of the major comorbidities leading to worse outcome in the context of COVID-19. So obviously, there's even more of interest to try to do something against diabetes. Now, in addition to the human suffering that diabetes causes, it has also emerged as a major economic burden. In fact, the global cost of diabetes was roughly 760 billion, that's with a B. And the cost of diabetes in the US alone in 2017 was 327 billion. And just as a comparison, in 2012, it was only, quote unquote, 245 billion. So again, you can see the massive increase in cost over a short period of time of just 5 years. In fact, the medical expenses for any person with diabetes are 2.3 times higher than for someone without diabetes. So obviously we are trying to do something against all these problems and trying to address them. And for that, we are lucky to have a UAB Comprehensive Diabetes Center that was made possible back in 2008, Interdisciplinary Research Center, or YORC as we like to call it. And as such, it has to have input from at least 3 different schools. But in our case, we actually have 250 faculty members representing all 10 different schools on campus, and with that, obviously, many different departments and divisions. Our work spans the full spectrum of basic translational, clinical, and outreach research. And our mission is to perform cutting-edge research and train future clinical and basic diabetes researchers. And what you can see here is the Shelby building. Our headquarters is up on the 12th floor when we're not staying safe at home. But obviously with that many faculty members, we're scattered throughout the whole campus. Now, what we want to talk to you today about are two major areas. One is to provide the very best care, and the second one is to identify better therapies for individuals with diabetes. And with that, I'd like to hand it over to my colleague, Dr. Ovalle, who will be talking a bit about providing the best care for diabetes. Fernando.
Right. Thank you, Anath. And good evening. I'd like to share with you a couple of quick updates regarding the clinical care of diabetes at UAB. One has to do with the telemedicine service that we provide. The biggest share of that is for diabetes. And another update is on the UAB Inpatient Glycemic Management Service that we've implemented about 10 years ago. We've been able to get very exciting data over the last few months. So first, the telemedicine services. As you know, UAB was actually very well positioned to the epidemic that we are facing right now, because thanks to the extraordinary work of Eric Wallace, the director of Telemedicine Services at UAB, we were pretty much prepared for what happened. We were not thinking about the pandemic of COVID, but we were just thinking that this is something that we needed to implement sooner or later. It happened that we had to kind of dive in all of a sudden. And as you can see there, this is the volume of our clinics. 70% of these is diabetes. And you can tell that the first two graphs, each of these bars is a week. The color gray represents in-person visits. The light blue is new visits, and the gray is return visits. So before COVID, the first 2 weeks of March, we were seeing, as you can see, a good number of patients there, about 20% of them new, and the rest return. And very suddenly, we had to stop seeing patients, or seeing a good number of patients. But as you can tell there, very quickly we recover. And we did for about 2 weeks, and by the third week and fourth week, we were back to the original volume. The bright blue color that you see there is phone visits. Then you start seeing a yellow and an orange color there, which represents the return video visits and the new video visits. As you can see, we quickly switched from in-person to telemedicine, phone, then video, and now the volume actually has been up, because video proved to be a very effective, very efficient way of seeing patients, especially those with diabetes. And now we've transitioned to what we think may be the way things are going to be in the future for endocrinology, at least for the diabetes care. I think we at UAB have a lot of patients who come from 2 hours, 3 hours away, and really coming here for diabetes services. It's takes half a day, if not a full day. And we think that the way you see things in the right side of this slide is the way things are going to be in the future. We're going to see probably 2/3 of the patients in-house, in clinic, but another third of the patients are going to continue to be seen through our telemedicine services. So that's just a quick update, just to let you know how quickly we embrace this at UAB and how things have transitioned and how things are going to look to be perhaps in the future. The care of diabetes is facilitated with the newer technology that you're probably aware, continuous glucose monitors and devices that are being used out there. So now on the inpatient side, we've also implemented a couple things. First of all, about 10 years ago, we were asked to improve the diabetes management in the hospital, and pharmacists as well as physicians from multiple specialties. And we put together a team of nurse practitioners, which has grown to about 5 of them, who focus all their efforts in diabetes care in the hospital. And we developed a set of standardized protocols. And we developed protocols for pharmacy and protocols for our MPs and protocols for primary care teams throughout the hospital. And over the last year, we felt that there was a need for perhaps a slightly different team of people who included the highly specialized team of endocrine fellows and endocrine faculty that could see some of the tougher cases and some of the cases that perhaps need a little different level of care. And we call this the fixed team, is the faculty fellows glycemic service. And this has now focused to see patients in the cardiovascular surgery services, cancer services, transplant services, and neurosciences, as well as other complicated patients using concentrated insulins or people who are pregnant, etc. So this, and if we can move to the next slide, has allowed us to, this is over the last 9 years, almost 10 years of data, bring down the number of severe hypoglycemic events. Then we phased in these protocols and services. And by 2015, we had most of what's now our glycemic management program in place. And we've been able to keep the blood glucoses much better control with a lot less variability, less severe hypoglycemia. And in the next slide, you'll see on the other end, also less severe hyperglycemia, down by 63%. So this is basically just a snapshot of what we're doing for diabetes care, clinical care at UAB. And thank you.
Great, thanks. So if there are any questions right now, obviously there will be time for questions at the end, but if there are any immediate questions for this clinical approach, we will be happy to address them right now.
Yeah, there is one question that was passed in. They had an uncle who had been diagnosed with adult diabetes for years, and he is constantly keeping his insulin low. He died in January of ALS. Are these issues connected, or is his ALS totally unrelated?
Probably unrelated, I would think. It's ALS. It's a condition of unclear etiology. And there is not a really direct link between diabetes and ALS, as best as I can, we can go to hypothetical scenarios, but I think for the most part, I would probably say there is no direct link between those two conditions.
Got it.
Okay, so we'll move on, and then if people have questions later on, we can address them. So I'll move now more into the mechanism and the molecular biology of things and how to develop new therapies. And so just as a reminder, how is our blood sugar controlled? As you know, we have the pancreas in our body, in the abdominal cavity, and in that pancreas, we have these islets that are the source of the major hormones that control our blood sugar. Insulin, or C-peptide, which we often measure because that's a measure of the insulin that is being produced in the body rather than insulin that is being injected, and glucagon, which is the counter-regulatory hormone that increases actually glucose levels, whereas insulin keep our glucose levels down. So as mentioned before, another major goal of our UAB Comprehensive Diabetes Center is to identify better therapies for individuals with diabetes. Now we may ask why. I mean, we're getting reasonably good control, and as you just saw by my colleague, we are making good strides also in providing better care, but there's still a lot of gaps. For example, there's still no oral drugs available for those people with type 1 diabetes or T1D. So this is typical. In the old days would be called juvenile diabetes, but again, it can affect people at any age. There are also no drugs that target the underlying islet dysfunction. As I showed you, those islets are primarily affected both in type 1 and type 2 diabetes. And so they overall produce then in the end too little insulin and too much glucagon. And there's also beta cell death. So beta cells are the cells that produce insulin. And still, even though we've known that for years, there are no drugs available that target exactly those issues. So how are we going to go about this? So for one, we obviously have to identify novel drug targets, because what we've done so far, we have medications that, obviously insulin that replaces the missing hormone. We have medications that try to push those beta cells to secrete more of the insulin. But again, none of them underlying, for example, the beta cell death. So to make a very long story short, we identified a detrimental protein that's involved in this diabetic islet dysfunction, and this protein is called TXNIP for short. It stands for thiodoxin interacting protein, and you understand why we prefer to call it just TXNIP. To address that, to kind of target that protein, we took two different approaches, and I will kind of describe both of them to you tonight. One is to try to repurpose an old drug, and we thereby used verapamil, which is a blood pressure medication. And then we also developed new drugs and developed a small molecule inhibitor to TXNIP, and I'll show you some of that in a moment. So first, in terms of the verapamil, we initially showed it in cell culture, and then we looked in different mouse models, and then we looked in human islets, and we saw that verapamil was able to inhibit TXNIP expression in those islets. And then we went ahead and did a randomized placebo-controlled trial in subjects with type 1 diabetes, and saw that those that were receiving verapamil, shown here in red, had a much better, did not increase, had much better glucose control, and with that required less insulin over time as compared to the placebo group, shown here in black. And it's kind of known that over time, this is over the course of 12 months, the requirements for insulin go up in newly diagnosed subjects with type 1 diabetes. And as you can see here with verapamil, it's not totally flat. They required a little bit more insulin also, but it was a lot less, significantly less than those that received the placebo. And importantly to note is that this did not go hand in hand with worse glucose control. As you know, the hemoglobin A1C is a measure of overall glucose control over the last 2 to 3 months. And when you look at that, you see that the lower the better, the subjects that were on verapamil had better control overall. It didn't quite reach significance, but overall they had better control as compared to the ones on placebo. And then very important, obviously, for people living with particularly type 1 diabetes, is that subjects on verapamil had much fewer hypoglycemic events, so fewer episodes of low blood sugar. And as you may know, those are very dangerous. They can be life-threatening and potentially lethal if the blood sugar drops too low and no one is there to help the patient. So that was very exciting to us to see that just by targeting TXNIP, and in this case with a known drug, verapamil, we were able to achieve such dramatic effects in terms of outcome. So what are the implications of this verapamil trial? While we cannot make general recommendation for the use of verapamil for diabetes, since it is not approved for this indication by the FDA, it has been FDA approved for hypertension for over 30 years. And physicians can prescribe it to the patients on a one-on-one basis, as it is readily available. And it's really been shown to be well tolerated and relatively safe. In addition, we have shown with the study a proof of principle that targeting TXNIP has beneficial effects, not just in different animal models, but also in humans with diabetes. And so obviously that gave us the courage, I guess, to embark on a small molecule screen. And we did this in collaboration with Southern Research Institute here on campus. And we screened over 300,000 compounds for drug discovery of a more specific TXNIP inhibitor. And what I'm going to try to show you right now is a very short, roughly 1-minute video, but it really is the result of over 10 years of work.
One of the most important sources of energy for our bodies is glucose. Our liver and pancreas work together to keep levels optimal and have us running in tip-top shape. In diabetes, this balance is disrupted and glucose levels become hard to manage. Normally, when glucose levels rise from something like eating food, islets in the pancreas release insulin, which helps tissues in our body take up glucose from the blood. On the other hand, when glucose levels drop, islets in the pancreas release glucagon that stimulates the liver to release glucose. In diabetes, there's too little insulin and too much glucagon action, fooling the liver into releasing more glucose and preventing the tissues from taking it up. This makes blood glucose rise even further and increases levels of a toxic protein called TXNP, which can lead to various problems, including fatty liver. A new preclinical drug discovered at UAB in Southern Research lowers toxic TXNP in human pancreatic islets and controls diabetes in animal models better and in a different way than existing drugs. Given orally, it dramatically lowers glucagon levels. It prevents unnecessary glucose production from the liver. The result? A better control of glucose levels than the best existing drugs now on the market. As an additional bonus, this drug also reverses fatty liver. So even though this drug isn't ready for human use yet, it does have a lot of promise for the future of treating diabetes.
All right, so here you can see kind of what you saw in the video, again, briefly summarized. We recently published this in Cell Metabolism, kind of one of the important journals in the field. You see here the small molecule that lowers glucose-induced TXNP expression. It's acting on the pancreas, and then there's less glucagon secretion from the pancreas that leads that lower glucagon levels in the serum leads to less production of glucose in the liver, and with that work against the development of diabetes. And then on top of it, this has shown to have protective effects on the islets and on the liver. And here you can see kind of, I'm going to go a little bit more in depth to show you what we saw in terms of the data and what was some of the things that were mentioned in the short video. What you can see in that first panel is a comparison of in red, again, the small molecule inhibitor, in black, the control, and then in blue and in the magenta, 2 known drugs for diabetes. One, the blue is the metformin, and in the magenta is the empagliflozin. So one a very established drug, the other one a newer one. And this is the glucose, and you see on the y-axis is the blood glucose level. Those are diabetic mice. So when they receive any one of these drugs with the metformin or the empagliflozin, their blood glucose does better than the ones that just receive control, shown in black. However, with our new small molecule, the glucose control is even better than any one of the currently used medications. On top of it, our small molecule was the only medication that was able to also not only improve glucose control, but also lower serum triglycerides, so serum fat, which was exciting. And so, better glucose control and lower blood lipids. On top of it, you can see that in this case, they were able to maintain higher insulin levels, even though in this mouse model, the beta cells were being destroyed. And their serum glucagon levels were dramatically lower. So that is obviously exciting. Again, kind of what you saw in the video, that's something that so far none of the currently available medications is able of doing. So with that in mind, we tested the small molecule in a model also of type 2 diabetes, and you can see here, those are very high glucose levels those are, again, this is a mouse model that is very severely obese and also diabetic. And within 3 days, after just giving that small molecule in the drinking water to these mice, their blood glucose levels dropped and remained low for 4 weeks. And they tolerated it really well. And as you can see, they did not affect weight. They actually optimized when they age over the 4 weeks, they gained more weight. And they gained equally more weight, so they were not sick or anything from the drug, but their diabetes was gone. Now, how did the small molecule do that? Again, we did kind of very extensive studies, but we're able to show that it is due to the lowering of the hepatic glucose production, so less production in the liver of glucose. And one major issue of several of such drugs in the past has been that they may improve glucose control, but they cause fatty liver. So when we looked at the liver of those mice, we saw that in the regular, those obese diabetic mice, they already had very bad fatty liver. But then that fatty liver was completely reversed in the mice that had received the small molecule. So not only was the small molecule correcting the blood sugar, it was also reversing fatty liver, suggesting that it may also be working in that context. So what are some of the advantages of this novel small molecule drug? I mean, for one, I showed you it's effective in models of type 1 and type 2 diabetes, and it's bioavailable orally, so it could be just taken by mouth. Again, as I mentioned to you before, currently for type 1 diabetes, people have to inject insulin or be on a pump. There's no other medication that can be taken orally for that. The mechanism of action of the small molecule is very different from any available diabetes drug. I showed you it's affecting hepatic glucose production and glucagon levels, which none of the currently available drugs do. By saying that, it also means that it could be combined with currently available drugs because they would act on a different mechanism, and so we could have additive effects. And I just mentioned the lowering of the glucagon levels and glucose production in the liver. So with that, the small molecule would also be effective in the face of insulin deficiency. On top of it, at least for now, what we've seen, this new drug does not cause any hypoglycemia, does not cause any hyperlipidemia, so no lipids in the serum, and no fatty liver, and no weight gain. So it's neutral from that end. It also specifically targets TXNP, and so the approach that has already been shown to be successful in humans, as I showed you with the verapamil trial. And this is an important point because I'll be the first one to admit that, especially in the field of type 1 diabetes, there have been many therapeutic approaches that have been kind of touted as a potential solution to the problem, but once they've been translated into humans, they were not as effective. Now, there's obviously no guarantee ever, but in this case, at least we know that the mechanism and the pathway and targeting this pathway is successful in humans. So we're a little bit more hopeful in that context that we could translate this into a successful therapy for patients with type 1 and type 2 diabetes, and also potentially with fatty livers. And so with that, I'd like to end and take any questions that may have come up. I wanted to leave enough time for any discussions that may have. Unfortunately, we can't do it in person, but hopefully you can type them in and we can try to answer them.
You bet. And we do have some questions already coming through the chat box. Let's go ahead and start with question. Would somebody who has had type 1 diabetes for over 50 years benefit from verapamil?
Possibly, and the reason I say possibly is that there's a big myth out there that people with long-standing type 1 diabetes would have lost all the beta cells. Obviously, the verapamil treatment is particularly working on trying to preserve beta cells. So once the majority of the beta cells have been lost, we would not expect it to be very effective. However, we've seen that even after 50 years, really, I had examples like that. Even after 50 years, there's still beta cell function there. And so in that context, it could help. For that, I would not obviously start it until knowing how much beta cell function is still remaining.
So what if a person with diabetes took verapamil? Would they not need insulin? Because the cost of insulin can be very high, and this whole thing would be great for low-income households.
No, it's not. It's not a replacement to insulin. However, we believe that if successful, it can lower the requirements for insulin, and I showed you that. So even though, especially in the context of type 1 diabetes, it would not, patients with type 1 diabetes require insulin. There's no way around that. But they would require less insulin, and the earlier you start, the less insulin they may require. So it would still be able to hopefully lower cost. And in fact, even from a healthcare perspective, we believe that this might be a good approach. Again, as you mentioned very rightfully, the cost of insulin is skyrocketing. So this is a way to try to lower the amount of insulin being required overall, but it is not a replacement. It has to go hand in hand with insulin.
So does all of this work with any CCB or only verapamil?
That's a very good question. We've only tested it with verapamil, and the reason we did so was that we initially looked at other calcium channel blockers, and verapamil was the most effective in lowering TIC-SNP. However, others like diltiazem were effective as well, and so we assumed that some of that would work also. There have been several studies, follow-up studies, epidemiological studies that actually support the effect of verapamil more in the context of type 2 diabetes. I had no time to discuss it now and to show the data, but it's been actually very exciting. The most recent one is one out of Taiwan, where they used their health record data. They have over 40,000 patient years, and they saw that the risk of developing type 2 diabetes was significantly lower in subjects that were taking verapamil.
So a couple of years ago, one of our attendees was on a lot of metformin. They got dehydrated, and that combined with the metformin caused a kidney accident. Is this hard on the kidneys?
No, it is no problem with verapamil for the kidney in that context.
Okay. So what about nasal insulin?
I can let Dr. Ovalle talk to that too, but my approach to it, I'm not a big believer in this, because the problem is that it's the accuracy. As I told you, I mean, one of the challenges that we face in diabetes is to try to mimic the very nimble and very accurate effects of the beta cell to balance any change in blood sugar levels and to secrete just the right amount of insulin. And we have trouble doing that even with the most sophisticated algorithms and the most exact insulin pumps. Now, the moment you try to dose something nasally, the accuracy suffers terribly. You know, you can have the sniffles and you're not absorbing it. And so when you're talking about something that is so important for life sustenance as insulin, it is tricky to dose it through the nose. But I'll let Fernando take more if he wants to add something to it.
I don't really have anything to add. She just said.
So what are the pros and cons of Trulicity?
Pros and cons of Trulicity. Well, Trulicity is a relatively new drug. It's belongs to a class of drugs that have been in the market for probably about 2005, so that's 15 years. So it's a relative. There is still things that we don't know very, very long term, like we do for metformin or for other drugs. But what we have learned so far in these 15 years is that it seems to be very effective at lowering the blood glucose. Perhaps more effective than a lot of pills, not as effective as insulin though. But it's associated to other, sort of side effects that we actually kind of like, mainly weight loss. It's one of the few drugs that we use for diabetes that allows patients to lose weight or at least not gain weight. while getting better glucose control. It does have some, nausea and some other undesirable side effects, but for the most part of no consequence. It also has, it's one of two classes of drugs that have been able to show that, of anti-hyperglycemic drugs that have been able to show that they have an effect on lowering cardiovascular risk, and actually also improving the kidney, the outcomes from the kidney perspective. So it's a relatively new class of drug that it has a lot of promise, and it acts in a very physiologic way, and we like it.
So will long-standing diabetics be good candidates for the small molecule drug?
There could be, yeah. It shouldn't. I think overall, the sooner you intervene, the better, because those drugs, both referral and the small molecule, one of the major effects is trying to preserve beta cells. Now, luckily, with the small molecule, it has the additional benefit that it lowers the glucagon and the hepatic glucose production, which is independent of the beta cells. So that's why, unlike the referral, I believe it would have also more effects in a longer standing subject with diabetes. But overall, obviously, the ideal thing would be to diagnose early and treat early, because you preserve more beta cells, and the more you have beta cells a subject has preserved, the better the outcome and the better the control.
Would referral have an effect on someone with relatively normal blood pressure?
Yes, and it has. I mean, because it does not work through blood pressure, and this is actually a very good question. That was our major concern initially, because we were treating, those were otherwise healthy young people. I mean, they were adults, but they were young, with referral, and we were concerned that their blood pressure may drop. I was sneaky. We were thinking the study was blinded, but we're thinking, well, we monitor the blood pressure very carefully, and maybe we're able to tell who's getting the drug. But we were truly blinded. There was absolutely no difference in blood pressure. So it's very interesting that referral, it lowers blood pressure when the blood pressure is high, but it doesn't lower it when it's normal. Obviously, you shouldn't start it, at a high dose very quickly, but we tapered it in, and we had no problems with that at all.
Does the UAB Comprehensive Diabetes Center accept new patients?
Absolutely. It's actually the patients that go through the clinic, but yes.
Yeah. So yeah, we have a very active diabetes clinic. We probably see about 8,000 patients with diabetes per year, and we still have we have new faculty members that are looking for new patients all the time. So we welcome patients with diabetes. Now, there is a lot of diabetes out there. We can't take care of all of them. So we have to, select some of the tougher ones, or some of the ones that need us the most.
Definitely understandable. So can you speak on the safety and side effects of the small molecule?
Yeah, so we obviously at this point, we don't know in humans, and this is the next big step that we have to, it has to undergo all the safety testing, so we could use it in humans. We've done already quite extensive testing in mice and also some in dogs, and it looked very, very safe. In fact, we were not, as you do some of these studies, the toxicity and pharmacokinetic studies, you're trying to escalate the dose till you see a toxic effect, and we escalated that far and to the max and did not see any toxic effects whatsoever. So overall, it really looks like it's very well tolerated, and we did not see any toxicity in any system that we looked at.
These are fantastic questions. Keep them coming in the chat box. We have some more to get to. Would these treatments help a type 2 insulin resistant person, because they produce lots of insulin, but they can't absorb it?
Well, they can absorb it, but it doesn't function properly in the periphery. And one of the problems actually, and we're actually studying this a little bit more, is exactly the effect that it has on the liver and the hepatic glucose production. So that aspect, obviously the beta cell aspect would be less relevant in someone with primarily insulin resistance, But the glucagon lowering and the effect on the hepatic glucose production would be major in that case, because one of the organs that is actually affected by insulin resistance is the liver. So unfortunately, even though the insulin is high and in general would block the liver from producing more glucose, since there's already high glucose levels, in someone with insulin resistance, the liver is resistant to that effects of insulin and just keeps on pushing out glucose, more and more glucose, despite the fact that they have already high glucose levels. So this, in that case, would be very helpful. So yes, we believe that that would be useful. Also, in that one mouse model, in the obese mouse model that I showed you, those mice are very, very insulin resistant, and it showed very good effects.
Since this is all pre-human so far, is it likely to get to the human stage? When does it get to the human stage? And can it help in time to not have to take insulin shots?
Well, those are very good questions. I mean, in general, drug discovery takes a long time. And as you can see, even with an approved drug like verapamil, it took us about 10 years to go from the target discovery to the actual clinical trial. Now, in this case, the target is known, and we know how it works and all that type of stuff. So at this point, it's a matter of getting, again, through the regulatory aspect and through the initial studies, phase 1, 2, and 3. Those are very, very expensive studies, and we're actually currently looking for partnerships to pursue those studies. They're beyond what we can do within the small academic realm, but we're looking for business partners to push this forward. But yes, we're hoping to get this as soon as possible into humans and to develop it, absolutely.
So does the new drug help reduce A1 levels?
Hemoglobin A1C?
All it says is, does the new drug help reduce A1 levels? So if somebody wants to elaborate on that in the chat box, we'll go back to that one. What age are patients when they switch to you from pediatric?
Typically, over 18. Some come until they're 20, 22. Some come a little earlier. It really depends on what else is going on, what's happening in their life, what's school-wise. Are they going to college? Are they not? Are they, sometimes they get pregnant. If they get pregnant, they send them to us earlier because pediatricians don't like to manage patients who are pregnant. If there's been a break in communications with the pediatrician, sometimes they tend to push him a little earlier. If they get along very well, and they tend to keep them a little longer because they have a good relationship. So there's a lot of factors that go into deciding the timing of the transition. But anywhere, I would say between 16 to 22, that's when they come over.
And let's double back to that previous question. Does the new drug help reduce A1C levels?
We would expect so, because we obviously did not measure. It's typically not necessarily done in models, in mouse models and other models, but it affects glucose control dramatically, and based on that, we would expect that A1C would be much improved.
Would a referral be needed from a PCP?
Not always. I think for the most part with diabetes, we pretty much know why they're here. So we don't necessarily always require a referral. We like the referrals because if there are specific questions they want us to address, that's great. But we welcome anybody who needs help.
So why did you choose to compare metformin and a SGLT2, but not GLP-1 or DPP-4 inhibitor?
We wanted specifically something that is not based on insulin secretion, because this is a model that has, this was an STZ model, the model is based on destruction of the beta cell. So it wouldn't make sense to compare it with anything that is trying to produce insulin, since the beta cells are being destroyed. So we're actually looking for something that could work in that context.
So I'm going to give you a very heavy question, and it may not even have an answer, but do you think there will ever be a cure to diabetes?
You want to answer?
Well, it depends on how you define cure. because and it's very interesting, and I've heard this. I had to kind of learn this just listening to people suffering from the disease. I think people have different perspectives about what does a cure mean. Does it mean that you give a shot and you are cured and you never have to think about diabetes and everything is like you never had the disease? Probably not. Okay. But can you have a treatment where you have a normal life and you don't have to think about it every minute of your life? Yes, absolutely. So I think that's kind of what again, it depends on how you define it.
Good answer. So does the UAB Comprehensive Diabetes Center help their patients with pharmaceutical patient assistance programs if they can't afford their medicines?
Our institution has a financial office that helps many of our patients with. Because of financial status of the institution, we see patients with no insurance. They have to register, they have to go through a process, but we do see a certain number of patients that have either have what we call they're underinsured or they have no insurance. And, all people who have no drug, they may have insurance, but they don't have any prescription drug coverage. And every drug is now very expensive. So we send them down to our financial office where some of our social workers work with them to figure out if they qualify for certain programs. There are public funds out there, there are private funds, there are drug companies most of the times have a program where they can, if the patient qualifies, they will provide the drug for free. And so we help them that way.
For somebody that wants to support the UAB Comprehensive Diabetes Center, where do they go? How do they help?
They should contact, and I probably should maybe share the screen again, if I can do that. Let's see. Oops. To go to Christian Smith, or they can contact me directly. And we have the development officer, and there are many ways to help. We're also happy to provide additional information if someone wants to visit or have one-on-one meetings with any one of us. We'd be happy to accommodate that as well. Let me see if I can pull that up somewhere. Okay, and here's the address, the email address for Christian Smith at uabc.edu. That's her phone number. Or this is also our website. You can look stuff up there, and there's also site giving that gives you all sorts of information, the different ways that you can support us. And again, feel free also to contact me directly, and we'll be happy to tell you more and tell you how you can be involved and partner with us. It's a very good question because, as I mentioned at the very beginning, we really depend on it, and we've grown a lot as you probably imagine over the last 12 years or 10 years, especially since I've been here. This has been primarily possible due to philanthropic support from the community. So it's been really great having a community that is so involved and working with us.
Go ahead and keep that slide up. We have a couple more questions here, but just in case people want to jot down that information.
Absolutely.
So is it possible for the Comprehensive Diabetes Center to accept patient from outside of the Birmingham metro, say Montgomery?
Yes, we do that all the time. As a matter of fact, I would say that, well, I don't have any hard data with me. More than half of our patients come from more than an hour away or 2 hours away. So.
And should patients with pre-diabetes see an endocrinologist?
Pre-diabetes. They don't necessarily need to. I think the primary care providers are, for the most part, very qualified. We're happy to see them. We're happy to guide them and counsel them. But it's probably a stage that they don't necessarily need an endocrinologist or diabetologist.
Any closing thoughts from either of you before we say goodnight and let you have your evening?
Well, I just want to thank people for coming tonight. I'm impressed by the number of people who showed up at this time of the day. So I thank you and appreciate your support. Yeah,
I just want to echo that. Thank you very much. And really, those were outstanding questions. So thanks for your interest, and thanks for listening in and joining in.
Dr. Shalev and Dr. Ovalle, thank you so much for joining us for tonight's webinar. Fantastic insight, background into what we're doing to combat that disease here at UAB. Thank you so much for being here.
Thank you.
And be sure to join us for upcoming virtual events. On December 3rd, we will welcome Dr. Rita Jablonski for Happy Holidays, Creating Dementia-Friendly Environments and Traditions. On December 15th, we'll explore the world of Hamilton with Dr. Brian Steele as part of Hamilton the Musical, the webinar. Then on January 21st, we will host an AI and libraries as we find out how artificial intelligence holds the potential to revolutionize libraries and information work. You can register for those and check out what else we have going on at alumni.uab.edu/events. And be sure to follow us on social media. We can be found at UAB Alumni on Facebook, Instagram, and Twitter. Stay on top of events, contests, and information. You can also find us on LinkedIn. There you see it, groups/102797, or just search UAB Alumni Career Community. Also, listen in on UAB Green and Told. Our podcast features members of the UAB community with new episodes releasing every other week. Dr. Donna Murdaugh and Dr. Andrew McDonald were featured in our latest episode. Hear the couple's story on Spotify and iTunes, or visit alumni.uab.edu/greenandtold. Finally, if you want more information about the National Alumni Society, visit us at alumni.uab.edu or simply email alumni@uab.edu. Once again, thank you so much for joining us for tonight's presentation with these two fantastic doctors who are doing great things over at the Comprehensive Diabetes Center. Have a great evening and go Blazers. Everybody take care.